Good morning, giant audience. Thank you for coming. Today's speaker is Abigail Uehling, and she's going to talk about the biggest thing that we had in our series of 7 talks for the 6 talks that we normally have, because in mid-May I always panic and I don't have enough speakers, and then suddenly I have 7. So thank you for being our 7th speaker. She's from Philadelphia, and she's going to talk not about neutrinos, not about microbes or germs or anything, but she's going to talk about the Red Sea and her diving experiences there, and I'm jealous. Thank you, Abigail.
Okay, great. Well, thank you all for coming, and thanks for having me. I'm excited to be here, and hopefully show you some fun pictures, and talk a little bit about um, my research. All right, so yes, yeah, let's see. Okay, there we go. So the focus of my talk today is going to be about a bioblitz in Oman, and then some of the Arabian Sea Star stories that have come out of our work in this region. And I thought I would start with a bit of background about myself. As Walter mentioned, I'm from Pennsylvania. I'm from right outside of Philadelphia. And for my undergrad degree, I went to Hamilton College in upstate New York, where I majored in biology. And then after undergrad, I took 2 years off of school and I interned at the Smithsonian Natural History Museum working in the invertebrate zoology collection there. And I enjoyed it enough that I decided to continue with research. So for my PhD, I've been at the University of Florida and specifically the Florida Museum of Natural History. My advisor is Dr. Gustav Paule, who's a collections or a curator in the invertebrate zoology collection. So, just to kind of give you a sense of where I work, sometimes people think of the museum as just the exhibits. So, trying to see if I can figure out the pointer. Okay, so Powell Hall is where most of the visitors are going to the museum. And this is where the butterfly exhibit is, if you've ever been there. But a large part of the museum is actually the special collections and Dickinson Hall, where we have all of the specimens for the collections. So we built this, well, not me, but the university built this brand new building recently called the Special Collections. And this is where most of the invertebrate zoology collection is held because our collection is in ethanol. So the fire marshal did not like the old building, and we have gotten a brand new building recently. All right, so I thought I'd give you a little bit of an overview of what I'm going to talk about today. So I wanted to start with giving a background in natural history museums and why they're important. I thought I'd do a quick review of invertebrates. Then I'll talk about our BioBlitz work and show some pretty pictures, and then I'll discuss my research questions a little bit more, and we can end with any questions you have. Okay, so um, natural history museums are really important for many reasons, but at their core, they are a reference library for biodiversity scientists to be able to study diversity of many groups. So you can see here, I've listed all of the collections that are at the Florida Museum of Natural History. And I work specifically in the invertebrate zoology collection, where we have greater than 700,000 specimens. So tons and tons of diversity are held at our museum. And there's also paleontology collections, archaeology, and ethnography as well. So just there's a wealth of information here. So at the core, natural history museums are made up of specimens. So I've shown a picture here of what some of the typical specimens that I work with look like. They are kept in ethanol in jars. So this is to keep them stable for scientists to study them. And each specimen has a series of information that is databased along with the physical specimen. So one of the most important pieces of information is a unique identifying number that goes along with the specimen. But then there's tons of information that can be captured as well. So we in our database, we also have the scientific classification of a specimen, so specifically which species it might be, where a specimen was collected. So I pulled up, sorry, this specimen is from Japan, and we have the exact coordinates for where it was collected, and we might have information about the habitat where it was collected and other important details. So collections are used for so many purposes. I just wanted to highlight a couple uses here. So they can be used as a reference for an example of a species. So a scientist could go to a collection and reference anything from the shelves to compare it to something that they might have. They can be used to describe new species. DNA can be extracted from tissues from specimens and used to create phylogenies or trees that I'll talk about in a minute. We can also compare characters between different species. So you can imagine, using the bones of vertebrate collections to understand how these bones have evolved in different species. We can understand how species are distributed across an environment, and how far species range across this environment. And the list really goes on. So before I get a little bit farther, I wanted to just briefly explain how to read a tree, because I'm going to show a couple more trees in my presentation. And they're basically a branching structure that shows a representation of evolution from the past to the present. So the bottom of a tree, or the base node here, represents the past. And as we move forward to the branches, the tips of these branches represent current species. So you could imagine that the structure shows you how related two species are. So A and B here are the most closely related, and they are more distant from C, which has an more distant common ancestor. That's just a very quick overview there. All right, so we have talked about natural history museums, and I work in the invertebrate zoology collection at our museum. So I wanted to just highlight why invertebrates are important. So they are basically animals that don't have a backbone and no bones at all. So they don't have what we classically think of internal bones, but they might have external skeletal features. So that is their most basic definition. And they encompass around 95% of diversity in the animal kingdom. So they're just incredibly diverse and important as animals. They're found all over the planet, in the terrestrial and marine environment. And many of the major phyla this is a way that we classify animals. Many are invertebrates. So I thought I'd give you a couple examples of groups of animals that are invertebrates. So I study this group, which is Echinodermata, and it encompasses sea stars, my main study, sea urchins, brittle stars, up at the top here, sea cucumbers, and crinoids, which are down at the bottom here. They're defined by this really special radial symmetry. So they all have 5-part symmetry in some way. They have a water vascular system, which if you've ever seen maybe the tube feet on a sea star or a sea urchin, they're these little feet that come out and help the animal move around, and those are all regulated by water that's pumping through the animal. And then they also have a calcareous endoskeleton. Another major group are mollusks, which are known for, again, their bilateral symmetry. They have this ventral muscular foot that they use to move around. You might see snails using their foot sometimes to move along. And they also have a shell. So it's less obvious in some mollusks, such as octopus, but they do have a really small internal shell. Another group of invertebrates are annelids, which are worms, basically. They also have bilateral symmetry. They're segmented, and they have these small bristles called chetae that are used to tell different groups apart. So people who study annelids will look at these different patterns of chetae and be able to tell different species apart. And then the last group that I wanted to mention are arthropods, which a lot of people are pretty familiar with. They're also invertebrates. They have bilateral symmetry, an exoskeleton, jointed appendages, and a segmented body, which I think can be pretty obvious. Like if you're eating a crab, you really can tell that there's a segmented body there, and this hard external skeleton. All right, so back to what we do in my lab. So our lab works with invertebrate collection. So one of our main goals is to increase sampling of diversity. And in the process, we often uncover new diversity, so new species. And as we are finding over and over again that there are many species that are yet to be discovered in these groups. So I put a map up here just to kind of give you a sense of what's lacking in some sampling. So this is a genus of sea stars that I have worked with before, and you can see some areas of the ocean are super well studied. Down here, New Zealand has all these sampling points, but the vast majority of the ocean is pretty understudied in many groups. All right, so now I'm gonna talk more about my fun fieldwork in Oman to study invertebrate diversity. So I wanted to start with explaining what a bioblitz is, and at its core, it's really a mass sampling of the diversity in an area. So you could have a bioblitz. Even here at Okhamic, you could try to document as much diversity in the woods as you can. In our case, we traveled to Oman to study diversity there. But my lab also is involved in bioblitzes all over the world. So I've also gone to the UAE. We're going back to the Red Sea, and Guam recently, I think I mentioned. So yeah, we're doing this all over the world. Okay, so this BioBlitz specifically that I'll talk about is in Oman. And I picked this one because I spent the most time here. So I thought I could, give you kind of a good overview of what it was like to work here. So this country is located on the Arabian Peninsula. You can see from all of these hearts here how extensively we ended up sampling across the coastline here. And we were really, really lucky to experience a really beautiful country. There's mountains in addition to the ocean there. So it was a great time in addition to our work. So this is a little bit far away from Florida. So I thought I'd mention why we decided to do a BioBlitz here. So Oman itself, and actually a lot of regions around the Arabian Peninsula, are not very well sampled. So there's a lot of potential for new species in these areas. In addition, this is also a really environmentally interesting region, which I'll explain in more depth in a minute. And but maybe most importantly, my advisor, Gustav Pauli, got a grant from NSF to fund our field work here. So the Arabian Peninsula is divided into what we think of as kind of four distinct ecoregions. So we have the Red Sea, the Arabian Sea, the Gulf of Oman, and the Arabian Gulf. And this classification is kind of based on very different environments between these regions, as well as oceanic fronts, so currents and weather systems between these areas. And other scientists, specifically people who study corals and fish, have found really distinct breaks in taxa between, fauna between these different areas. So they find that what you find in the Red Sea might be very different from what you find in the Arabian Sea. And so knowing all these features, this seemed like a very interesting place to do more sampling. These maps here can maybe give you a good sense of how different environments are between these different areas. So there's temperature ranges up at the top with darker red colors showing higher temperatures. And you can see the Arabian Gulf up here is just super, super warm compared to the rest of this ocean. Salinity is also ranges a lot. Chlorophyll and phosphorus, maybe most in the Arabian Sea. You can see it's very red. And perhaps the best example of how this range in environment affects the ecosystems can be seen when you compare the Red Sea to the Oman coast. So the Red Sea is known for being pretty oligotrophic, which means there's low nutrients in the water. And you see a lot of these classical coral reefs. So these huge, huge coral structures that house a lot of diversity. And this is different from the Oman coast down here, which experiences a lot of seasonal upwelling. And upwelling is basically when nutrient-rich water comes up from the bottom of the ocean and brings the cold temperatures and lots of nutrients along with it. And so in contrast, in Oman, we see that coral doesn't grow the same way. It doesn't grow in these large structures. Instead, you see kind of small patchy communities next to kelp. So that's a little bit odd. That's not something that you see in many oceans in the world. All right, so our main work in this area took place in 2021 and 2022. But my advisor Gustav has been to this region and specifically Oman many times. And when we were here, we sampled three main localities. So Muscat, which is the capital, Masira Island, which is kind of midway on the coast, and then Salalah, which is in the very south western part of the country. And as I said, this was led by Gustave Palle, as well as Michel Clairbout, who is he's now a retired professor from the local university in Muscat. And it involved extensive sampling with many invertebrate specialists from all over the world. So here I'll show a few more fun pictures of the fun part of a lot of what we do is going out in the field. And so a typical day usually involved us getting on a boat and going diving. So we would look at maps and see places that looked like they would be good for diversity, as well as suggestions from locals who might tell us spots that they would suggest going to sample diversity there. We did a lot of diving, and we used a couple of different methods to collect diversity while diving. So in as a first pass, we could pick up large fauna that we might see on the bottom. So if an urchin is sitting there, that's a pretty easy thing to sample. But a lot of diversity is hidden or not very obvious to most of us. So we would brush rocks to collect really small animals that live on those rocks. A lot of snails are very small, worms are very small. We would also bring up, or so you can see me brushing a rock here. This was often my job because I was pretty new to some of these methods, and so they would say, "Abby, go brush the rocks. You can handle that." But I've learned a lot throughout this work. So another thing that we would do is bring up buckets of rock. So there's a lot of diversity that lives in the rock face itself. So again, it takes a lot of time to try to get this out of the rock in the water. So we would pull up the buckets when we came back from our dives and spend some time looking through them back in the lab. This is just a nice sampling of some of the stuff that we've found. Is a really beautiful environment. Here, this is Colcita. This is a pincushion sea star. It's very different, from kind of your classical sea star. On the bottom here, this is an Aquilinastra sea star. This is the group that I study that I'll tell you a little bit more about. But we just saw some beautiful array of colors in this environment. And then I had to put some more sea star pictures in here. This is a group of sea stars that seems to be mostly restricted to that island called Masira that I mentioned. And another one of the group that I study up here, this is a sea urchin right here that has these really beautiful bands and patchy coloring that is super distinct in this area. And then this is a nudibranch. So these are sea slugs that are very beautiful to find underwater. So in addition to diving, we would also collect things by hand picking them from the intertidal. So when the tide was really low, we would go out with our buckets again and look under rocks to see what we could find. There's a lot of animals that burrow into the sand. So we would sometimes use shovels to try to dig them out, or we would also use this is a pump. It's called a yabby pump that if you are trying to get an animal that's deep in a burrow, it makes it a lot easier to kind of suction the animal out. And yeah, a lot of shrimp are collected that way in these environments. All right, so once we collected our animals, we would bring them back to the lab and we would try to sort them as best as we can. We would try to keep them stable. So we could go through kind of our standard pipeline. And the first step to that is getting a really great picture of each animal. So we will always have that reference. Yeah, so you can see people are often really excited to come back to the lab and see what they've collected. All right, so after we bring the animals back and stabilize them, we'll do a couple of things. So we'll take a really great photo, as I mentioned. We will then also take a piece of tissue from each specimen for DNA sampling, and then each specimen gets fixed in ethanol for transport back to the Florida Museum. And so we would spend most of the day diving and then stay up pretty late at night processing all of our samples. And this, I don't think this number is quite right. I forgot to change it, but we have collected, I want to say, more than 10,000 specimens from Oman over the course of our field work there. And there are lots of cool sea stars. So I've been very excited. So one of the really important aspects of this is we give each specimen an ID, and that ID number stays with the specimen as it moves down our pipeline and eventually becomes part of the museum collection. And this helps us make sure that we know exactly where that specimen was collected and can use all of that information for future research. After we finish with our fieldwork in Oman or any place that we are doing a BioBlitz, we have to transport all of our specimens back to Florida. So you can see here, this is maybe our least favorite part of the whole trip, where we have to pour off most of the ethanol that we have used to fix the specimens, and then we put them back into bags, and we actually usually bring most of them back in our checked luggage. So all of those specimens can come back with us, and we have our permits in hand. So when we go through customs, they know that we've collected them with all the correct paperwork. And yeah, it's exciting when we finally get everything back safely. So after they come back to the museum, all the specimens get cataloged and put into our museum database. And a lot of the tissues that I said I had taken get sent off to extract DNA from these samples. And yeah, then the specimens join our collection. So you can see here, the process of making sure that all of these samples have been stabilized. And then they get sorted into our shelves where they are categorized based on their identifications, and scientists can easily come access them, maybe if they're visiting our collection. And we do get a lot of visitors from all over the world that come to study invertebrates here. So all of our data is available on the internet, and this is just to give you kind of a rough idea of what we're creating from all of our work. So if a scientist wanted to search our collection and figure out what we have available, they could easily do that. And all of those photos that we take in the field then become part of the collection database as well. All right, so I I mentioned briefly that a lot of our work leads to DNA sequencing, and um, essentially DNA sequencing allows us to figure out what diversity might be new and how species are related. So we sequence the DNA, and then we use phylogenetics to build a tree to infer unknown diversity. So what we find often is after we have done this, we find a lot of cryptic diversity. So this means that there are species that are so similar that you can't even tell them apart. But once you sequence them, this shows an example of a piece of a tree here, you can see that there's two different clusters. So there's a cluster at the top, oops, and a cluster at the bottom. And sometimes this is super surprising to scientists who have assumed that they know what they're looking at, and then they realize that there's more diversity there. All right, so I'm going to kind of briefly touch on some of my PhD work that has come out of our collections from the Arabian Peninsula. And as I mentioned, these involve sea stars. This is my main group that I work on for my PhD. And yeah, they're really pretty. There's so many colors and so much diversity in this group. All right, so back to the Arabian Peninsula. We had talked about a little bit earlier about how there are different ecoregions in this area. So again, these are distinguished by environmental differences, but some other work with other groups has shown that there does seem to be distinct species that exist between these four regions. And I already demonstrated, the difference between the Red Sea and Oman, but just to reiterate that point here, the environments are super different. One in the Red Sea is very rich in standard coral reefs, and one has these really patchy coral reefs and kelp communities. So you can imagine that this creates a different environment for other species that live here. So for one of my chapters of my dissertation, I guess multiple chapters, I'm working with this genus called Aquilinastra. And I'm interested in how this genus is distributed across the whole Arabian Peninsula, across those different ecoregions. So a little bit of background on this group is that they are super diverse. There are about 32 described species, 6 specifically known from the Arabian Peninsula. But they're really challenging to work with because you can't use very many features of them to tell different species apart. And one of the main features that you use is these spines that are on the surface of these sea stars. So there's a lot of pretty colors, in these pictures, but the colors don't really help. Instead, I have to put them under the microscope. An interesting feature of this group is that there's a diversity of life history. So life history is kind of referring to different aspects of how species might reproduce or how they exist in the environment. And in this group, we see some differences in how larvae exist in the water column after they have been spawned. So larvae could either be planktotrophic, which means they can feed actively in the water column, or they could be lithotrophic, which means that they can't feed in the water column. Instead, they have to rely on a single egg that sustains them until they can settle to the bottom of the ocean floor. So this photo kind of shows a contrast of these larval eggs. This really big egg is one that is lithotrophic. So the larvae will just rely on this egg and potentially not be able to travel as far because it can't actively feed. Another strategy that is interesting in this group is fissiparity, which is asexual reproduction. So a lot of species in this group actually kind of pull their arms off of their body and they can regrow a whole body from a single arm. And sometimes they'll also split themselves down the middle to do this. It's a pretty wacky strategy, but you can see we can observe the differences in individuals that do this. They have more than 5 arms versus individuals that don't only have 5 arms. All right, so I was really excited to have sampling across this whole region to be able to tackle some of these interesting questions in this group. As you can see, we've done fieldwork in the Red Sea and Oman, and all the way up to the UAE. All right, so one of my main questions was, what is the true diversity of this group? Are there hidden species that we're not aware of? Are species restricted to ecoregions? So to those 4 main regions across the Arabian Peninsula, as we've seen in other taxa? Why are or what factors are driving the species distribution? So why do we see these patterns? And then lastly, how are some of these reproductive strategies distributed across a phylogeny? So across an evolutionary tree, how do we see how these traits have evolved? All right, so to quickly answer my first question, what is the true molecular diversity of this group? So although there are 6 described species, we've actually found that there are likely 5 new species in this region. So after sequencing the DNA and building this tree, we can see how species are related. The species are noted at the side of the tree here, and we can tell that there's new diversity based on how the branches cluster. So that's really exciting. There's a lot of new diversity in this region, which was not super surprising, but still very exciting. So my second question was how species are restricted to ecoregions, as we've seen in other taxa. And these graphs or these maps, I'm sure are a little bit hard to read, but I can describe to you what we're seeing. So basically, we found that most species are in fact restricted to a single ecoregion. So you can see in this map, one species exists here, one species exists here, and the same happens over and over again, except for one species that is actually distributed across the whole or most of the peninsula. My third question was what factors are driving species distribution? So how do we see, or why do we see these patterns of species being very restricted to different ecoregions? And the answer for now is that this is research that is to come, kind of my next phase of my PhD, but I have a couple hypotheses. The first one is that they might be restricted by a limited ability to disperse across these ecoregions. And this goes back to the life history strategies that I was talking about in this group. So a lot of these species seem to not be able to feed as larvae in the water column. And so my thought is that maybe they're not able to travel as far, thus they are more limited in their distribution. And then my second hypothesis is that these species are restricted by environmental factors. So again, we've talked about these really drastic differences in environment, and maybe this is influencing restriction. And maybe it's also a combination. And my last question here was how are reproductive strategies distributed across the phylogeny? And remember, I was talking about fissiparity, which is basically fission in an animal where they pull an arm or a couple arms off, and then they can regrow their whole body from this. So I was interested on how the pattern of how it's evolved in this whole group. And what we found is that this strategy has evolved multiple times. So I've noted it with these red stars here. And you see that instead of evolving in one group, we see that it's happened 4 times across this group. So I find this to be really interesting because it's not the simplest explanation, but there must be something that is making these species turn to fissiparity in these environments. All right, so my next steps are pretty exciting. So I have probably 5 new species that I want to describe from the specimens that are coming back from this work. So that's really exciting, and it's also sometimes a long process. I want to continue to explore what's driving these restrictions across the ecoregions. I want to figure out why we have so many species that seem to be reproducing with fissiparity, so breaking their body and cloning it essentially. And then I also work with sea cucumbers and sea urchins, some other questions of diversity in these groups. So I'll keep working on those throughout the rest of my dissertation. And yeah, that's the end of my talk today. Thanks for coming.
See some questions. And as usual, I have the microphone, so I get to ask the first one. How do you feel a young, good-looking lady in an Arabic Muslim country? Did you feel safe?
Yeah, that's a great question. So when I decided to come to the University of Florida for my PhD, I did know that I would likely be doing field work in Oman. And that's something that I was definitely like, I've never been there. What is that going to be like? But I talked to a lot of folks that had been there before, and I can say I honestly felt very safe. I I think that it's a particularly special country in that a lot of Omanis are very respectful of foreigners coming in and are very welcoming. They're happy to share their country with other people. So although the majority of the population is Muslim, they respect that people have other beliefs, and so I think that, it was still recommended that you are modest in how you dress. But for the most part, people were very welcoming and very respectful. And I think also, yeah, very hopeful that as foreigners, we were having a positive experience there. Yeah. Well, that's a great question. Yeah.
I thought I read mine. Oh, I just wanted to tell you that I taught school on Okinawa for 11 years.
Wow.
And Okinawa was not a great big island. So the most interesting thing that we did was collect seashells. And I won't go into the how we did it and everything, but my seashell collection is over there. I gave it to what natural history here.
Nice.
So it's it found a home after all of that.
That's awesome. It's probably part of the collection I work in then. Yeah. 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 That's very special.
I just asked before, did you have to wear the black robe? Was it
Yeah.
was it in
so-
mosques or just in public or something?
No, so in Oman, most people don't wear one of, that extensive of a covering. Some people do actually because it's like a fashion thing too. They're definitely more covered up and they'll wear, a head covering. Usually that's like more brightly colored. And I brought a scarf just in case, but I wasn't expected to wear one. Just when I went into, they have what is it's called the Grand Mosque there. They expect you to wear a covering. Yeah.
Yeah.
Mhm.
I had to wear it there. That's why I was asking.
Okay. Yeah.
But my question is, I'm interested in this Fissiparix
Mhm.
species. One of the definition of two species is that they do not breed together.
Yep.
So how do you get different species with Fissiparus species? How, they are just reproducing, they are reproducing themselves.
Yeah.
So how do you get different species?
Yeah, that's a great question too. So, I guess in short, we yes, we do consider species to often be individuals that are, interbreeding with each other. So it looks like some Fissiparus species might also occasionally become sexually reproductive, so they might stop, cloning themselves and, take a break from that and go back to, reproducing as a norm, what we usually expect. So we kind of don't know enough about some of these species that are doing this to say that they only reproduce through fissiparity. But more insight would definitely allow us to, be able to say more confidently how these species are existing in these populations. Yeah. Mm-hmm. Yeah.
Do all sea stars, can they lose a leg and grow a new one? But does the leg that fell off grow a new sea star?
Yeah, that's a great question too. So, yes, some species can definitely do that. not all species. There are some sea stars that have this great regeneration ability. So if they lose a leg, they can regrow it on the original body, but the leg that fell off wouldn't necessarily regrow a new leg. But there are a lot of species that can do that. So I guess it's a species to species, yeah, dependency.
Two questions. One, excuse me. One is in Oman or here, do we have any of these live specimens that are being grown to see if they do interbreed and how they and their behavior, etc. And my other question is, if we wanted to see some of these, I know that museum from way back when I used to do some work. I'm a vertebrate zoologist, and I saw a welcome sign. So my second question is, how do we get to go in and see some of the?
Yeah, great question. So, I don't know of any active collection, like aquarium collections of these that are currently being studied in that way, like in the US. I think that in the Red Sea and some of these other regions, there's been scientists who have monitored the populations more closely, but none that I'm aware of doing it right now. But I would love to have an aquarium population of these to study a bit more, and that's something that I have thought about trying to make happen. But an interesting fact is that there are a lot of this group of sea stars that are pests in aquariums. So if you have a saltwater tank, or if you see a saltwater tank, sometimes you can spot them. They're usually kind of on the margins of the tank, and they're really tiny. They have, maybe sometimes 5 legs, but often, 3 or weird numbers. And sometimes it's hard to find them because aquarists like don't want them there, so they'll try to keep scrubbing them off. But I've seen them in a lot of tanks in Gainesville even. So yeah. And then your second question was oh yeah. Mm-hmm. Yeah. Yeah. So that's another great question. Oftentimes we have like groups that come in. So I could, see if you brought a group from Oak Hammock, for example, and contacted a collections manager there, that they're often very happy to give tours. Yeah, it's a big part of what we do. So yeah.
Um, I'm also very interested in this phenomenon of regeneration.
Mm-hmm.
I'm trying to figure out how it biologically it happens. So I imagine they have stem cells, a lot of stem cells.
Mm-hmm.
Is there research being done, and what exactly is the mechanism of this regeneration?
Yeah, that's a great question. That's something it's definitely like a little bit outside of like the type of research, right? But yes, I guess my short answer is they do have these special this ability to have cells that can very quickly regenerate in those areas where they've been harmed or are regrowing themselves. So I've I'm looking into kind of the genetics behind this a little bit, but it's a bit more maybe on the cellular side of research. So but there's a lot of research that's being done with sea stars to look at those cells. Yeah.
Have you done research or studies on the Oregon coast or in Alaska? Because they have these sea creatures too, and they're beautiful.
Yeah, I haven't personally, but I've been to the West Coast, and actually, checking out some of that the tide pools there and the coast there is how I got really interested in studying invertebrates. But my advisor, Gustav Palle, has worked a lot on the West Coast, and there it seems that there's always more to study. So I expect that our lab will be back there to do more research. Yeah.
I think it's interesting that I have heard that it's against the law to collect seashells now in Okinawa with the uh, that there have been some serious people that have tried to have a seashell collection, but I guess we don't gather again to do that. I just thought it's interesting that it's against the law with the Japanese.
Yeah, I guess it really depends on where you are sometimes, right? Like what regulations there are, and in Florida we have a specific permit to collect stuff all the time, but seashells that are washed up on the shore are a little bit different. So yeah, I think it just depends, but definitely shell collectors can be a really big part of like our research too. Yeah.
Abby, thank you for sharing with us. I'd be interested to know whether or not your area of the Natural History Museum has interests in the U.S. areas, like, for instance, U.S. territory in Guam or American Samoa or Virgin Islands or Puerto Rico. It seems like access and cooperation would be very easy to come by there. And the last thing is, will one of these 5 new species you're dealing with be named Abigail?
Well, I'll answer your second question first. I don't know that, people often name them after themselves. I kind of have to wait for someone to name one after me. But I've thought a little bit about names and stuff. So that's definitely a fun aspect. And sometimes when you choose a name that people recognize, then the species also gets more interest because people are like, oh, like for example, there's a shrimp that's named after Thor, the superhero, and people love to find that shrimp based on that. And then your first question. So yes, I guess we do have a lot of interest in working in U.S. territories. So there's we collaborate a lot with the Smithsonian Natural History Museum, and right now they have they work with the Bureau of Ocean Energy Management, which is interested in documenting diversity all over the U.S., including the territories. So that's really exciting. And then I think I had started to mention to you that I have a collaborator who's currently working in Guam, and he recently got funded by the naval base there to do some work on the diversity of the reefs. So yeah, so definitely it's a fun collaboration opportunity. Yeah.
I have a question
Yeah.
back to visiparity. I would imagine that this is basically what they call vegetative reproduction.
Yes.
So you would
exactly.
imagine that they may have a colony which is homogeneously identical versus those that were sexually reproduced, in which the genetic variability will be different from the vegetative one. Is that what you find? Different colonies depending on how they were reproduced? Is that what you find? Or they had different colonies of the same starfish?
Yes. Yeah. So I guess there's kind of a couple parts of this. We don't know if they only reproduce with fission, right? So there is a possibility that they might switch back and forth between reproductive modes. But what we do find is that a species that only reproduces sexually, that is always consistent, right? So then if you have a species that is vociferous, it's possible that they might go back and forth. But yes, in some populations where they're doing this, we can look at the genetics and see that they are, identical genetically. And then there's a little bit of variation, maybe sometimes geographically. You might have a slight difference from that population that is genetically identical. So yeah, there's so to answer your question, yes. And I guess we also kind of need to know more about how these populations are functioning, or how the species themselves are sharing genetic information. Yeah.
Also, an unrelated question. Did
Mhm.
you have difficulty getting alcohol to preserve your critters?
Yeah, that's another great question. Yeah, so we were working in a Muslim country, right? So alcohol was a little bit, carefully regulated, but we had a collaborator that was working at the local university, and so he had access to some ethanol there, and then I think we also ended up getting, yeah, I guess a lot of it had to be ordered ahead of time just to make sure that we had enough when we were in the field. Yeah.
Okay, my experience diving, so the sea stars were quite large in my experience. But you're talking about very tiny ones. Could you address the variation in size in the different species?
Yeah, so there's really quite a variation. The smallest species that I study are often the vociferous ones, and they can be like smaller than a dime, like really small. And then the pincushion stars that I mentioned earlier that are pretty really abundant in Oman are like this big. So there's really a range, and the small ones are definitely harder to study sometimes.
So when you mention the vociferous ones are by dropping a leg and regrowing it, is their DNA identical all the way down the line?
Most, well, yes, in sometimes within populations. So, for example, if you think of the whole species, maybe there's an island population where most of those individuals are identical, but there is still a bit of variation in the genetics in some of those species. Yeah.
But that's a normal variation based on some kind of external event that makes a slight modification.
We don't know. So it could be because they might go through periods where they are reproducing sexually, like maybe they're able to share, DNA, or maybe there's some kinds of mutation in part of the population, but that's a great question. Yeah. Yeah. Yep.
Any other questions? Well, this was wonderful. I am so glad you came, and it's one of the most exciting talks that we've had
Oh,
all
thank you.
summer. Thank you very much.
Thank you. Yeah. Yeah, thank you very much.